Hi there! Welcome to my poster. I'm Maxime Borry, a doctoral researcher at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, in this corner of Europe. Since I have the next 5 minutes of your time, let me tell you about my research on ancient DNA metagenomics. When we work in ancient DNA metagenomics, we usually deal with two different kinds of samples. Coprolite or paleofiscid sample, here on the left, that are these fossilized feces, and we use these samples to usually study the ancient gut microbiome, whether it's for humans or other animals. When we want to look at the oral microbiome, we can work with samples known as dental calculus, which is what happens when you don't brush your teeth. And luckily for us, ancient DNA researchers, toothbrushes weren't invented until relatively recently. And actually, using ancient DNA metagenomics data, you can do pretty much the same analysis that people do on modern metagenomics data. So here in these recently published articles that one of our collaborators published, she managed to reconstruct hundreds of genome using de novo assembly from sequenced coprolite samples. However, one of the characteristics of ancient DNA that you cannot avoid is the so-called deamination. Deamination is a process that happens almost systematically for ancient DNA sample, and this is the loss of amino group of cytosine, which then become a uracil and is read by sequencers, by DNA sequencers, as a thymine. So you will, if you look in ancient DNA articles, you will often hear about C2T deamination. So when you work in genomic with a single reference sequence, with a single reference genome, this is the typical analysis pipeline. So you start with DNA, do DNA extraction, library preparation, sequencing, and then you map your reads back to a reference genome, the reference genome that you're interested in. And since you're doing ancient DNA genomics, you will have to look at this so-called damage profile. What you can see here is on the x-axis, you have the position in the reads, and on the y-axis, you have the average proportion of deamination for each position of the read. So in this sample, on average, on the first base of each read, you would have around 22% of deamination. Or in other words, in this sample, if there is a cytosine at the first position of this read, in 22% of the cases, it will have become a uracil rather than a thymine. This was the case of genomics. However, when we work with metagenomic, and especially metagenomic de novo assembly, we potentially generate thousands of reference sequences, since each contig that we assemble is going to become its own reference sequence. So the workflow is pretty similar, still starting with DNA extraction, library preparation, sequencing. Then we go into the step of de novo assembly, and then we actually map all the reads back to all the contigs that we assemble. So hence this many, potentially hundreds of thousands reference sequences. And if you would follow the same process as your colleague doing genomics, then you would have to look manually at each of these damage profile. And whereby informatician, it's not really feasible. And even if it would be feasible, we're way too lazy to look at these plots manually for hundreds of thousands of plots. So we had to come with an alternative approach to automate this process so that you don't have to look at each of these plots. And this is the method that we propose, PyDamage, which does an automated ancient damage identification and estimation for contigs in ancient DNA de novo assembly. So how does PyDamage actually work? So in PyDamage, we model the C2T deamination damage for each position as a binomial distribution. So binomial distribution takes two parameters, n, which is the number of cytosine at a given site, and p, the damage frequency at a given site. In this toy example, we have a binomial distribution represented for each position, and PyDamage then try to fit two different models. A null model that assumes a uniform distribution of the damage parameter p, or a uniform distribution of the damage, to put it more simply, and a damage model which assumes decaying distribution of the damage parameter p, or a decaying distribution of the damage. What we then do is compare the likelihood of these two models thanks to a likelihood ratio test, and because we're doing this likelihood ratio test, we actually get a statistical estimation of how likely a contig is to contain damage. How to install PyDamage? It's relatively easy, since it's written in Python. You can just pip install PyDamage, or if you like more conda, you can also conda install it. How to use it? Also, again, relatively easy. PyDamage analyze. Here I want to generate plots, and I give it my alignment file. What kind of output do you get? So you get first a damage estimation, meaning you get the damage parameters of the model. You get a p-value. Here the null hypothesis of this statistical test is that the reference does not contain damage. And because here at the previous step we wanted to have a plot, you get this kind of plot where you can see the fit of the two different models compared to the real data. If you want to learn more about PyDamage, feel free to visit my poster at maximebory.com/PyDamage_poster and try to beat PyDamage. There is actually a game in the poster, and you might get some prize if you manage to beat PyDamage in the poster. Thank you for listening.